This is a production of Cornell University.
All right, thank you, Nina, for the introduction, and thank you all for joining me today for my seminar. Again, my name is Brandon Miller, and my presentation today is titled Hickories in Horticulture, Developing Novel Nursery Crops. Just a brief overview of what you can expect from this presentation. I'm going to go over hickories in horticulture, the reasons why horticulturists should value them, and some of the problems that we encounter with them in the nursery industry. And I'll do this, once I go over them, I'll talk about specific projects that hone in on some of these problems that we've encountered and finding solutions for them. And some of these projects I'll go over in a little bit more detail than others. And then finally wrap it all up with talking about the future research needs, what we need to continue to work on to improve hickories in the nursery trade, some citations, and of course my acknowledgments. Now, as Dr. Bassuk mentioned, my research that I've been working on here at Cornell has been investigating approaches to improve nursery production and transplant success, and also looking at our understanding of stress tolerance, mainly with hickories, but also with a handful of other valuable plants for use in managed landscapes. But today we're going to talk about the projects I've worked on specific to hickories, and just a little bit of overlap with something that I've worked on with birches and how those are tied together, how those connect. So getting started here, why are we talking about these plants that we might use in managed landscapes? Why are we interested in this at all? And really, I'm approaching this from the perspective that trees in our managed landscapes, trees in our urban forests are threatened by two main general factors, biotic and abiotic stresses. And in the most recent years, especially here in New York State, we've seen exotic invaders like the spotted lanternfly and the emerald ash borer, insects that threaten the stability of our plantings. And we also see more dramatic weather patterns, severe weather events, flooding, drought, that our trees have to be able to withstand all along the way. And our managed landscapes are particularly vulnerable when we have limited biodiversity. So the species that compose our managed landscapes, if we don't have a lot of biodiversity, they're threatened by this. And when that's the case, if we do get that specific pest that favors that type of tree or the conditions that threaten those trees, then this is what we end up with. And so we want to avoid that. And the way we do that is to bolster biodiversity and look at other trees that we can add to our plant palette as options for biodiversification. So one of those groups of plants that we would like to consider is or are the hickories. Now, when I say hickory, species that belong to the genus Carya, many people probably are thinking about shagbark hickory. It's one of the more iconic species. It has exfoliating plates, exfoliating bark, beautiful golden fall color, and edible nuts. It has a pretty broad distribution, and so people from different regions may often think of this species. But what people don't often realize is that the genus Carya is more diverse than that. There are approximately 19 different species, depending on who your preferred botanical authorities are. And the genus is broken up into three groups, three subgenera: the Pecan hickories, true hickories, and the Asian hickories. And everything that I've put in this red rectangle here, these are all found, they're all endemic to North America. Only one of them is restricted to Mexico. All the plants names that are in bold there, those can all be found in the United States. They're endemic to the United States. And so we can actually look at the distribution of all of these species on the generic level. If we look at the United States on the county level, so with the counties that are highlighted in green, those are counties where one of these species of hickory can be found. And you can see it's a very broad distribution. I've drawn this red line through, so everything to the east of that red line, those are native populations, and everything to the west are likely naturalized populations where people have moved these plants around and they've naturalized in those locations. But in the eastern United States, basically that whole half of the United States there, we can see that there are species that range from the furthest northern conditions to the furthest southern conditions. So we have genetic variation that can accommodate cold climates, warm climates, coastal ecosystems. There are niche species that inhabit niche ecosystems with upland sites and wet riparian habitats. So there's a lot of variation in this genus, and that means that we have many traits that we can use to our advantage in horticulture. And I'm mainly looking at these plants from the ornamental perspective or their use in managed landscapes, but other horticulturists may value them for their nut production or their other, timber products or other components. But there are lots of reasons why we want to look at them. When we're thinking about the ornamental traits and some of these useful components and factors with hickories, one part that I like to talk about is the bark, because shagbark hickory, one of the more iconic species, has that exfoliating bark, but not all of the species have that. There's a broad range of characteristics that we find in the genus, scaly, flaky bark like we see with pecan, or fissured bark with some of the other species. The autumnal display is really something that many horticulturists really favor with hickories. They have a golden fall color, and they have some of the best golden fall color of any temperate species that we can find. And so these are a handful of taxa here with more golden yellow or almost the amber color like we see with pignut hickory. I mentioned site adaptability, and if you look at the different species in this genus, you'll find plants that are on upland sites, some sites that tend to be more dry. We see them in mesic sites, somewhere in between. And then, like I mentioned, there are plants that inhabit these niche riparian locations, rivers, streams, marshes, and sometimes in swamps. There are species that are adapted to those locations and growing habitats. And if you look in reference books, if you ever look up hickories, oftentimes terms like resilient will be used with them, and there really isn't a whole lot of evidence for it. But we think of these plants as adaptable, and we suspect that they're very resilient. And when we do see them in stressful locations, although rare, when we do find them, they seem to do particularly well on sites that we would often think of as challenging for most trees. I'd be amiss not to mention their fruit production. So they do produce edible nuts, all the species do, but some are preferred more than others. And pecan, being a member of this genus, is a massive nut crop, not only in the United States, but across the globe. And so many of these species are favored by people for their edible nuts. And these trees are also, the nuts in particular, are consumed by other organisms, not just humans. Because they're well adapted to the entire eastern half of the United States, they're integral components to various habitats, and their ecological significance is very important. So we should value these plants not only for our personal use, but for their role that they play and the ecosystem services that they provide. And so you may be wondering, well, if these trees are so great, why are we talking about them today? What's the problem? And like anything else, these trees are not perfect. There are some issues in terms of nursery production that we could improve upon to make these plants more viable nursery crops and more accessible plants for use in managed landscapes. And the three main groups are transplanting, propagation, and general production. So there's some kind of issue with these areas that we need to address. Oftentimes, hickories are associated with their strong dominant taproot. It is true that hickories do have a strong taproot that persists beyond their juvenile phase. And because they have this taproot, many horticulturists, there's this generalization that they cannot be transplanted because they have this strong taproot. And that's something that we're trying to find evidence for, determining if that's true or if that's not true. But it is a generalization that's made and common among nursery growers. And really, you can see that taproot development shortly after germination. Hickories, for the most part, are produced by seed. And right after germination, you can tell they put a lot of energy into the taproot, and that investment in the taproot persists for their entire juvenile phase. And that juvenile phase can, it can vary from species to species, but it is multiple years. Ultimately, they put so much energy into that taproot that the shoot, the above ground portion, is often neglected. And so from a nursery grower's perspective, if you have these teeny little sticks in pots, it takes a long time to have a saleable plant, and it's hard to make a profit. So nursery growers might shy away from them just because they are so slow to grow in their juvenile lag phase with their episodic shoot development. And because they're grown from seed, when growers do put in the time and energy to invest in these plants and put them in production, we end up seeing a lot of variability. So you might find plants like we see on the left, where they have a strong apical dominance, a nice form. These plants are 6 years old at a nursery here in New York. And that plant on the left is saleable, but the two plants on the right, same age, 6 years old, have formed more of a shrubby habit. And they're not the same size, and they're definitely not saleable. So this is an investment that many nursery growers would shy away from because you can't have that after 6 years. So naturally, then you would question, well, why don't we asexually propagate them, have a more uniform crop? And while asexual propagation is possible, the techniques that are used mainly for nut production, they're just not amenable to nursery production for managed landscapes from an ornamental perspective. It could be that there's graft incompatibility, or it could also just be that because bark is one of those ornamental traits we're interested in, these top grafted unions just are not pleasing to the eye, and they're just not quite suited for our scope. So how do we take these plants that we see on the left, the small little shagbark hickories, and how do we end up with a nice saleable plant that is more common in the nursery trade and then more available to practitioners? I have a series of projects that I'm going to go through. The first three, I'll go through just a little bit more detail with some brief data, and then I'll just use pictures to kind of describe the rest of the work that I've been doing to try to tie all this together. The first project I want to talk about is something that we did trying to predict drought tolerance, trying to characterize drought tolerance of hickories. I mentioned that sometimes they're referred to as resilient. And so we want to actually put a number to that, determine where they fall relative to other trees. And our hypotheses were first that the species we studied, the riparian species, might actually be more drought tolerant than the mesic upland counterparts. The reason for this is if you go to some of these riparian habitats, you'll see that there's seasonal drought that many of these habitats will go through. And so we wondered if perhaps these plants are already adapted to the fluctuations between inundation and drier conditions. And our second hypothesis was that the degree of drought tolerance would be variable within the species. So again, we're trying to quantify and characterize drought tolerance. We studied a handful of species useful for northern landscapes and 2 interspecific hybrids. And then we're just trying to get a better idea of how variable this trait is within and across populations. So the species that we studied, bitternut, pignut, kingnut, shagbark, hickories, pecan, and then 2 interspecific hybrids, Dunbar's hickory, which is shagbark crossed with kingnut, and Nussbaum's Pecan, which is a pecan hybridized with the kingnut hickory. So we have a handful of taxa. And the data that I'll present today, these data were collected in 2019 and 2020, so they're all averaged over 2 growing seasons to get a better idea of the variability from year to year. And we collected samples in the spring and then in the summer for both years. All the plants are located in Ithaca, New York, with the exception of 1 population of Carya liceniosa, which was in Moravia, New York. And we have 9 observations per tree per season. We can see the number of trees that have contributed to this data set. And the methodology for how we did this was vapor pressure osmometry with corrections according to Schumann et al. And I'll talk about that on how we did this on the next slide. Data were subject to a one-way ANOVA, and we used Tukey's HSD for mean comparisons. Okay, so vapor pressure osmometry, what is it? How are we doing it? Well, the idea here is we're going to characterize drought tolerance using the leaf water potential at the turgor loss point. So that's the value that we're trying to predict. To get that, we need to first get the solute concentration of the leaves in the spring and in the summer. So we start by going to the site and finding our plants that comprise this data set. And we collected stems from those plants in this growing season. And we looked for leaves that were plants with leaves that were not afflicted by disease. They weren't fruiting or flowering, so they were standardized. We removed them and cut them underwater and take them back to the laboratory. We left them in the laboratory covered overnight. And again, they're cut underwater to remove embolism from the stem. And the goal there is to let them get back to full turgor. So the stems are left to equilibrate overnight. The following day, we would come back, and in image number 3, you can see that we use a 7-millimeter cork borer to get a leaf disc. The leaf disc is frozen in liquid nitrogen to break down cellular barriers, and then we feed that into the vapor pressure osmometer, which gives us the solute concentration value. Solute concentration is then fed into Van't Hoff's relation. So we can calculate the osmotic potential, and in this case, full turgor, because the plants were allowed to equilibrate. And then we take advantage of this more recent work that's been done by first Bartlett et al., which they looked at a global data set and created an equation for predicting the leaf water potential at the turgor loss point using the osmotic potential value. And then we use a refined equation that is looking specifically at trees that belong to the temperate region so that we have more accuracy. And that equation was developed by Schumann et al. And so I have 3 slides of data. I'm going to show, I'm going to walk through what this, how you decipher this figure, and then that'll kind of tell us a little bit more of what the results were and how we've characterized drought tolerance here. So what you're looking at are means of the predicted leaf turgor loss point of the species in hybrids. So that turgor loss point value is negative megapascals, is on the x-axis. You can think of it, the more negative the value, the more drought tolerant we think that that plant would be. On the y-axis, we have our species, and the species are ordered in descending order according to their summer means. So the summer means are represented in the black bars, and spring means are represented with the gray bars. The overall, across all the species and all the plants, means are represented with those 2 vertical lines for summer and for spring. So I just want to look at the black bars today. I want to look at those summer means. So these are the means for that predicted leaf turgor loss point for all these species. And what we can see is that Carya glabra is up at the top. That is our least drought tolerant species. And if we look at the bottom, our most drought tolerant was Carya liceniosa. If we look at those species and where they've kind of segregated here on the y-axis, we see that our riparian species are mostly at the bottom of the y-axis, and our upland mesic species are mostly up at the top. Now, this scale is relative, and if we had looked at some of the other data that was presented by Schumann et al., they've done this with various trees that we might use in the urban environment. We could take, for example, something like Acer platanoides, the Norway maple, a plant that's really tough and is a very good urban tree. And if we looked at the mean for that plant in summer, we would find it right around -3. So relative to other trees that we know to be very drought tolerant, all of these hickories are exceeding that, but there are species that seem to be better than others within our data set. We can look at the same general idea, but using 2 separate populations, so plants of known provenance. And here we have plants of Carya liceniosa, the kingnut hickory. It was the species that had the greatest drought tolerance overall in our last slide. And if we look at plants of known provenance in the Arboretum versus plants from a wild population in Moravia, we can see that provenance matters and there's some variation there. We can also take this a step further and look at our species, Carya glabra, the one that was the least drought tolerant of all of our species. And we can look at the number of individual plants that contributed to the data set. So on the y-axis, you have the number of individual trees that we looked at, and we can see that there's quite a bit of variation just within that single population of plants. Mainly plants number 1 and 8 on the bottom of the y-axis, when we're looking at those black bars, seem to have a greater capacity for drought tolerance than do plants 4, 10, and the other ones up at the top. So there's a lot of variability even within the populations. So the conclusions that we can draw from this are that of the taxa that we studied, Carya liceniosa was the most drought tolerant. Generally, our riparian species, at least the ones that we looked at, were more drought tolerant than the upland taxa, again, within this data set. Provenance is, of course, important, and there's a significant amount of variability even within populations. And so we think that this means if we can find a good way of asexually propagating these plants, that clonal selections could be made that would be well suited to drought-prone landscapes. On the note of asexual propagation, that is one of the projects that we looked at. So we trying to look at something other than top grafting, finding a way to asexually propagate hickories. And Dr. Bassuk and the Urban Horticulture Institute developed a modified-stool-bed-layering technique that she used for her hybrid oak program. And so we thought, well, we could try this and see if that works for species of Carya. The goal here with this experiment is determining if auxin concentration, exogenous auxin, has a role in rootability of the plants. And so the species that we're looking at are Pecan, kingnut hickory, and Shagbark hickory. Our treatments are, again, those concentrations of IBA, so 0, 4,000, or 8,000 parts per million. And then we have 9 single stock plant replicates per treatment combination. And I'll go ahead and I'll walk through what this process looks like on the next slide. So for those that may not be familiar with a mound layering, or specifically this modified version of it, this is how we do it. So we start with plants that are established in the field. So you can see that on the far left. These plants were planted, let us to establish for a year, and then we came back the following spring, and we coppiced them. We stooled them or cut them back. We let those plants bud out. They formed adventitious buds. And once they reached kind of that stage that we see with the picture on the right, we covered them with a light excluding implement. In this case, we just used pots that we had covered with foil. And the idea here is to exclude light so that those shoots will etiolate. Once those shoots have etiolated to about 10 centimeters tall, which we see in the middle there, we then apply exogenous auxin. And this is where we had either the 0 parts per million, 4,000, or 8,000 parts per million auxin concentration. And we applied that to the base of those etiolated shoots. We then mound those plants. So we put soilless substrate along, or we mound it up around them. And basically you want to cover where we've painted the base of those shoots with auxin, where we hope roots will arise. So we cover that with a soilless substrate. And then we transition those plants back to full sun because they are etiolated. We don't want them to burn. And so we transition them with a shading implement for approximately 2 weeks. Now, we did this on a plant-by-plant basis. If this was being implemented on a larger scale in a grower's stock plant field, maybe the same clone would be in one row and they could tarp all of them at once. But again, because this is a research project and each plant was a seedling, they're all at different times. So we did this on a case-by-case basis. So you can see all of the plants here in the field at the Bluegrass Lane Research Center. And those plants are allowed to grow throughout the summer, and we revisit them in the fall. So these plants we revisited in October, right around when the leaves had senesced. We uncover the mounds and look at the individual shoots, which are called layers. And we look to see if any roots have formed where we had applied our auxin, or for the control, just ethanol. And then those layers can be removed from the stock plant. They can just be cut away. And that leaves you with rooted layers and a happy graduate student. So you can see, of course, a rooted shagbark hickory here with a phenomenal root system. So we can look at the data here with this table. The columns of this table from left to right, we're looking at the different taxa that we studied, the auxin concentration, the percentage of rooting on a stock plant basis, and then the mean count of roots per layer, a value kind of determining the quality of rooting. So if it rooted, how well did it root? How many roots did you get? So we can look at the first 2 species, Carya illinoinensis and liceniosa, that's pecan and the kingnut hickory, respectively. And we can see that with 0 parts per million auxin, 0 parts per million IBA, we had no rooting. With 4,000 and 8,000 parts per million, we did have rooting, almost identical responses for those species at 4,000 parts per million. But with 8,000 parts per million, we had a higher rooting percentage and a greater number of roots per layer with Carya liceniosa. If we compare that to Carya ovata, again, the control resulted in no rooting at all. Interestingly, the 8,000 parts per million also resulted in no rooting, but 4,000 parts per million was the sweet spot for that species. And we had 95% rooting and 11.6 mean count of roots per layer. So a really phenomenal response with the shagbark hickory in that situation. So what do we take away from this is that the exogenous auxin in this setting was necessary for rooting. The concentration is important, and it seems that the different species respond to that concentration differently, not just in rooting, but also like we noticed with shagbark hickory at 8,000 parts per million, a lot of those plants died. So we think that that auxin concentration was phytotoxic. The overall conclusions and what we can take away are that the modified stool bed layering technique can be used to root species of Carya, and that asexual propagation techniques like this open up opportunities for clonal selections and basically alternatives to top grafting. Worth noting, the roots of all of these plants were fibrous. They were not coarse, like a seedling of a comparable size. And so we question if perhaps this root morphology has implications for some of the other generalizations and problems with these trees that we see in nursery production. The other component that I mentioned was that the plants, when grown from seed, are slow to develop in the above ground perspective. So we wanted to determine if plant growth regulators could be used to modify shoot growth, in this case, shoot extension. And so we looked at gibberellic acid. We wanted to find out if gibberellic acid 4+7 could influence shoot extension of some hickories. And then we wanted to characterize it and determine, is this actually something that could be useful for nursery growers? So the species that we studied here are bitternut, pignut, shagbark, kingnut, and mockernut hickories. And then we also have pecan. The treatments, there are 3 treatments. They were either the control, which was untreated, or we had 500 parts per million GA4+7 dissolved in ethanol. So we have a GA solution at 500 parts per million. And the 2 treatments there, it was applied either to the apical meristem, which we called the bud treatment, or it was applied to the basal region of the shoot. These plants were already 2 years old coming into this study, and treatments were initiated in their 3rd growing season. So right after they were taken out of the coolers for their winter dormancy, the buds were brought into the greenhouse. The buds became swollen, and then they broke. And that was when we started to apply GA4+7. The way we did it was we just used a cotton swab in the ethanol solution, in the GA solution. And we, again, applied it either to the bud, the apical meristem, or the basal region of the shoot. And we did this at 3-day intervals for 27 days. We had 11 single plant replicates per species in treatment. And the experiment lasted 160 days from start to finish. I have a main effects table here looking at 2 factors that I want to share with you today: shoot extension and caliper. Shoot extension, we saw that the factors, there was an interaction between species and treatment. And then for caliper, the species level was the only factor that was important. So there was species variation for caliper, but the treatments had no effect on caliper. And I want to start there with a specific figure to look at the caliper. So again, these are seedlings. And so on the x-axis, we have the different species that we studied. And on the y-axis, we have caliper in millimeters. Again, they're very small seedlings. And we're looking at caliper pooled across treatments because species was the significant factor there. And what I want to convey here is that there's variation across these species, that if we were to only look at one species that we're familiar with, like Carya ovata, and conclude that all the other species must be very similar to that, well, we'd be wrong. And so that's the point that I'm trying to make here, is that each species should be considered on a case-by-case basis when we're talking about production in the nursery and use in horticulture. They each are a little bit different, and we shouldn't generalize across the genus. Now back onto the GA4+7, how did those treatments influence shoot extension? On the x-axis, we have the species here, and then on the y-axis is shoot extension in centimeters. Within each taxon group, within each species group, from left to right is the control, stem treatment, and the bud treatment. If we look at the 3 species on the right-hand side, Liceniosa, ovata, and tomentosa, we can see that those treatments didn't have any effects. If we look at Carya glabra, the species second in from the left, we can see a trend, but once again, nothing to write home about. However, if we look at Carya cordiformis and Carya illinoinensis, so bitternut, hickory, and pecan, we can see that bud treatment resulted in a very significant amount of shoot extension. And so we can look at those plants in particular. On the left is bitternut, hickory, and on the right is pecan. Within each picture, control, stem, and bud treatments. And again, that bud treatment, the plants almost, they follow the graft pattern identically. And so you can see the shoot extension is rather important and large here with these 2 species in particular. So what results and conclusions can we draw from this? Well, that bud treatment with GA4+7 resulted in a 234% increase of shoot extension for bitternut hickory and a 144% increase shoot extension for pecan. Neither of the treatments, whether applied to the bud or to the base of the stem, influenced caliper for any of the taxa. And the two species that did respond, may be worth noting, both belong to the same subgenus. So they're both considered the pecan hickory group. And maybe that says something about the mechanisms that are actually controlling the episodic growth and the shoot development. It's worth noting that we tried this experiment again with plants that had already flushed their growth for the season. So in the midsummer, once they had set a terminal bud, we tried this exact experiment and we were unable to induce a second push, a second flush of growth in one season. And that was the original hope. But this is all kind of a jumping off point for determining how we can use plant growth regulators to influence plant growth in the nursery. Now, for the last component here, I'm not going to go over any data. I'm going to use some pictures to kind of tell the story. In the last experiment, we were looking at smaller plants that might be useful for specialized growers who might already be selling like a trade gallon pot. But what about for everybody else? People that nursery growers that would be looking at larger material and the larger material that typically practitioners expect for use in the landscape. And we wanted to look at how hickories and pecan, how they grew and how they survived containerization and field planting. So if you were a nursery grower, if you were growing plants in a container nursery or in the field, you would purchase in what we call liners. So usually they're bare root, they're smaller. These were whips, they're 12 to 24 inch tall plants. They came from a forestry nursery. They're harvested from a field with a U blade in the winter. They're shipped in the winter. And then in the spring, you would either pot them into the containers or you would line them out in the field. And so we designed an experiment looking at 4 different species to examine how did those plants survive both of these processes. And we also wanted to look at 2 different types of containers, the standard black plastic and one of the new grow bags, the root maker. So we're looking at number 10 or trade 10-gallon sized pots and then plants that are lined out in the field. Now the plants in the field, I'm not going to show an image of those because they were just so small compared to the plants that we had in containers. The plants in the field have barely grown over the 3 years that we've looked at them. And whereas the plants in the containers were leaps and bounds, there were big differences. And so I have some images here. On the bottom row, we have the plants in the grow bags. And on the top row, we have the plants in the standard black plastic. The 4 species that we studied from left to right are bitternut hickory, pecan, kingnut hickory, and shagbark hickory. And immediately what's apparent is that there's a difference in the robustness of the plants in the bottom row compared to their plastic pot counterparts above. And so we thought that was interesting, but what might again be more interesting is the fact that the shagbark hickory, which is the plant on the far right in both rows, they're much smaller than their congeners. They're much smaller than their relatives that we've looked at. And this picture was taken at the end of year 2 of looking at these plants, and this was in the fall. So the shagbark hickories had already senesced their leaves, but you can see they're basically still a stick in a pot, whereas the Carya cineosa, pecan, and bitternut hickory are much larger. And so again, this theme of this recurring theme of drawing conclusions based on a species we're familiar with for the whole genus doesn't apply. And so we think that in this case, the bitternut hickory and the kingnut hickory could possibly be close to a saleable size for a number 10 pot product. And this would only be a 2-year investment for a container grower. So that was the end of year 2. And we had this kind of strange phenomenon in year 3. The plants had broken their buds and they started growing well, but then for our pecans and our bitternut hickory in particular, we started to notice some really strange symptoms that we couldn't really attribute to any kind of management or cultural practice. So we ruled out herbicide drift. We ruled out our, we were fertilizing all the plants as we have done for everything else. But again, the bitternut hickory and pecan developed these really strange symptoms where the leaflet would curve. We had a necrotic margin, and in some cases, the plants would rosette. So this kind of has most people scratching their heads, but this is where a side project I had been working on the year prior comes into play, where we were growing yellow and sweet birch in 2019. These are plants that are Again, underappreciated in horticulture and not common in the nursery trade. Dr. Bassuk wanted some for campus so that we could teach in her plant materials course, creating the urban Eden, yellow birch and sweet birch. And so we decided to grow some on our own. And in year 2, growing these plants in soilless media, we started to see these strange symptoms: curling of the lamina, necrotic margin, and it turned into rosetting, or kind of described as looking like a witch's broom. So in 2019, we looked into this problem. We looked at the literature, and we found that these symptoms were consistent with something called mouse ear disorder. Mouse ear disorder, at least in the scope of the nursery world, is only really documented with pecan and river birch. It occurs with other plants, was originally found with soybean, but for woody plants that we would find growing in nurseries with soilless substrates, these two species are notorious for exhibiting mouse ear disorder. So we can see the symptoms on the right-hand side. And what is mouse ear disorder? Mouse ear disorder is essentially a function of nickel deficiency. So when we're normally talking about nickel, with nickel being a heavy metal, we're usually talking about it in the sense of phytotoxicity or too much. But for a handful of plants, and it seems mostly these are ureide transporting species, they require, possibly they require more nickel, but they definitely require some. Nickel is a necessary nutrient for plant growth, but these plants seem to require a little bit more. The reason being is that nickel is an essential cofactor for urease activity. Urease activity will decline when nickel is not supplied in adequate amounts, and urea builds up in the tissues, and that's what is thought to result in the curling of the leaves, the rosetting, and the necrotic margin. So nickel nutrition turns out to be very important for some of these birch species. And at the time in 2019, I had reached out to John Reuter at the University of Georgia, who had done this work in the early 2000s with river birch, figuring out what the problem was and how it's alleviated. And so we collaborated with him to determine if the commercial product, Nickel Plus, which is a supplemental nickel product, could be used with yellow birch and sweet birch. Right now, the label is made for the label is for pecan and river birch. So we did this experiment, and here we have yellow birch on the left, sweet birch on the right. And from left to right within each image, you have the control twigs, which are symptomatic, and then 4 twigs that have been treated with nickel. And you can see with these birch that the nickel alleviated the problem. And so this work will be published this year. And ultimately what this did for us was give us a leg up in figuring out what was probably also happening with our hickories. The symptoms were very much the same. And as I said, in the literature, pecan is notorious for having this issue. So now we are looking through the other species of Carya to figure out which ones may also be susceptible. And this general nutrition component will be important for nursery growers in developing this crop. And we did see it, like I said, mostly on bitternut hickory and on pecan, but we've also found it on most of the other species that we've worked with. And so this work is ongoing, and our preliminary trials have suggested that that is exactly what's going on, that this mouse ear disorder, which we originally found with our yellow and sweet birch, also is happening with our hickories. So that work is ongoing. The other project that I'm not going to be able to talk in detail about today is ongoing, and we're collecting data this spring to wrap this project up, is the effects of species size and production technique on the transplanting or transplantability of 4 species of Carya. We were really lucky to get this experimental crop of shagbark hickory from Schichtel's nursery, and you can see them here either in bare root with the plastic bags or balled and burlapped, and they're large plants. When I spoke with the company, these plants, I think, were in the ground for 14 years to get to this size. So we really lucked out in timing. And Dr. Bowerly has been helping me with looking at different ways of measuring new root growth with those in-ground soil cores that we see there. And ultimately, we're looking at root hydraulic conductance and general growth responses to try to figure out how those effects of species size and production technique affect transplant success. And I have an image on the right of Dr. Bassuk in Ryan, Georgia, We were admiring a tree that we'd planted earlier last spring, and I really appreciated their help because this was in the early stages of COVID when everything was still rather complicated, but we were able to safely continue with our research. So we hope to have those data at the end of this year and published, and so hopefully we can contribute to that area of transplanting, and that area of the problem with hickories. And looking forward, so how do we take all this information that I've mentioned so far and continue to develop hickories as crops from either my perspective or the perspectives of other people in horticulture and beyond? Well, we of course can refine and modify the techniques that I've talked about today under the categories of propagation, production, and transplanting. This is really a jumping off point. I think we've made some really good progress, but some of these techniques can be modified for becoming more applicable to other nursery settings. Those that have interest in selection and breeding could really take advantage of an opportunity here where we have so many species, so many unique provenances, and so many traits that could be bred for and cultivars, clonal cultivars selected for. And mainly I'm thinking ornamental traits, which is obvious, expanded application. All these trees produce nuts, and we could look at the variability in nut size. Usually for nut production, you want a larger fruit that's easier to crack and taste good. But maybe for some of our urban examples, we might want a smaller fruit size or less fruit that's produced on an annual basis, so that they aren't considered a nuisance in some situations, maybe for high traffic urban areas. We can look for plants that are suited to production. I had shown the images of the variability in the field at Schichtel's Nursery. And so we could have cultivars that are just more uniform, clones that we could have more, like I said, uniformity and less variability in production. And when I talked about the drought tolerance, we talked about how some plants, individual plants, seem to be more stress tolerant in the way of drought stress and drought tolerance. So we could be selecting plants from that perspective as well. And then I want to mention also germplasm and conservation, that botanic gardens, arboreta, and public institutions like the USDA germplasm repositories play an important role in not only preserving the genetics that we have, but in preserving the beauty of these plants going forward. And with any luck, with some of this information and with a little bit more future research, plants like this Shagbark hickory will become the norm. They'll be a lot more popular in a park, street tree planting, or maybe even in your own backyard. So I'd like to share here just the sources that I've talked about, some of those literature sources, and then some web images that I've borrowed or modified. And before I finish up, I just want to acknowledge some institutions and some people. Of course, the funding organizations, McIntyre, USDA, NIFA, McIntyre Stennis, the Horticultural Research Institute. I'd like to thank the institutions that have housed this work: Cornell University, Cornell CALS, and SIPS. We work with John Reuter with the Birch work, and he's from the University of Georgia. There have been industry collaborators with all of this that have been very helpful. NIPAN provided Nickel Plus, the nickel supplement to us. Schichtel's Nursery has been very helpful, and Jim Kisker in particular saved the day in the early days of COVID when I couldn't travel to the nursery to acquire samples, and he really helped us out a lot. Jean Grace, Pride's Corner, Dr. Bowerly for helping with the in-ground soil cores. The research assistants and technicians have been incredibly helpful: Hazel, Eileen, Jimmy, Nina Fanfang, and Ryan Georgia. And last but certainly not least, my research committee and my advisor, Nina Bassuk, has made this work possible, and I can't thank her enough for all of her help. And like I said, just making this opportunity available for me to study this area that I'm so passionate about. And Dr. Mark Bridgen and Dr. Tim Setter for their help. They've been incredibly helpful and supportive of me in any way that they can throughout my PhD. So I'd like to say thank you to all those people. And there are other people for other projects I haven't talked about, but you all know who you are, and I appreciate all of your help. And with that, I would just like to say thank you to all of you for listening to this presentation today. And I'd like to go ahead, and if we have any time left, take any questions.
Brandon, may I start out with an easy question for you?
Sure.
I thought your findings using the vapor pressure osmometry were very nice. But I do have a question about the spring versus summer
Sure.
measurements.
Sure.
And I'm wondering how long after leaf expansion were those specimens in the spring taken? Was it very shortly after expansion, or was it after a few weeks?
Yeah, that's a great question. So these were taken, so it was, you could consider it mid-spring. The reason I chose mid-spring is that that was at the time that all of those plants that I studied, they had all completely expanded their initial growth, at least the first few leaves of their initial growth. So it was shortly after expansion, but not too close to bud break, if that makes sense. So I think it was late May or the first week of June is when I had done it here. So they had fully expanded, but those first few leaves had fully expanded.
Would you say that they had fully greened up, or were they still in the greening up phase?
Yeah, they definitely they were fully greened up. I went more towards fully developed than less developed. And the idea there was that we could get a baseline once they had fully expanded to compare what they were fresh for the growing season compared to after they had endured the conditions of the growing season in the summer.
Great, thank you.
Thank you.
Looks like there's a question in the chat.
So the question I'm seeing in the chat is that they think they've seen the mouse ear disorder in persimmon also. Is this something I've come across? And it happens when they use less aged pine fine growing media. Yes, it is definitely possible to happen to other plants, and I have seen it on persimmon that I've grown myself. I had seedlings of persimmon that had the same issue, and we treated it with Nickel Plus, and that worked as well. So there are other plants that definitely could have this problem, and I know for a fact that persimmon is it's not documented, but is definitely something to think about. The literature suggests it's plants that are they tend to be ureide transporters, and also a lot of these plants tend to be riparian, or plants that you'd find in more wet locations. So I'm not sure if that's always the case, but it might be useful for predicting which taxa could have this problem.
This is Frank Schroeder from Chemistry. I'm
Hi, Frank.
Here because I actually grow hickory trees a little bit as a hobby, and I really enjoyed learning so much more about them. I have one comment and one question. So I have one tree in my yard of 30 trees that clearly has the mouse ear disease, and I've wondered for years what that is. And I'll try a nickel treatment, see if it changes things. It's a pretty big tree, not big, it's a medium-sized tree, maybe 6-inch diameter or so. And I noticed that they really respond to nitrogen a lot in the young trees, the grafts that I've planted, and maybe reflecting your results as well. So pecan and shellbark or kingnut, they really respond very well to nitrogen. You can get 2-3 feet of growth in a year now.
Right.
But the ovata, the shagbark, it's the slowest. It's always the slowest. Even though once they're large, I have older trees that grow 2 feet a year, but the young ones just won't.
Yeah, the juvenile phase is definitely a part of it. They're the absolute slowest when they're juvenile, but once they kind of reach that past that juvenile phase, there seems to be a switch. And like you mentioned, the kingnut or shellbark, hickory, and pecan, once they get to that point, then they'll put on multiple feet of growth in one shoot expansion, one season. And I agree that they do appear to be heavy nitrogen feeders as well. There's another question in the chat from Ken Mudge about why we think the riparian species would be more drought resistant than the upland species. It's counterintuitive. Drought tolerant is what we're aiming for here. And yes, it is counterintuitive. But when you look at, like I said, when you look at some of these riparian habitats, sometimes they are more inundated and wet throughout the year. But some riparian habitats, they're on the edge, and they go through a fluctuation between inundation, and sometimes they'll go through seasonal droughts. And in swamps, that actually tends to be the case. If you look at like taxodium in the wild, sometimes a year they're a couple feet underwater, and sometimes a year it's completely bone dry in the middle of the summer. And so there's an other area, body of work that's been exploring this, that for some, well, although it's counterintuitive, that those plants are actually sometimes better adapted to fluctuations. And so that's why we wondered if they'd be better suited to drought. And the plants that we looked at from mesic sites aren't necessarily locations that get really, really dry routinely. Like those weren't species that we would find on like rocky outcroppings, for example. I think that if we target other species that might be on really dry sites, that, you know, we'd find evidence that those plants could also be very drought tolerant. But again, the idea with the plants from riparian habitats is we might have two birds, one stone. They could be capable of tolerating the spectrum back and forth between flooding and drought.
Brandon, first of all, terrific seminar and really well-developed research. So congratulations on that.
Thank you.
Given that the vegetatively propagated, etiolated plants showed a more fibrous root system than do seedlings, might they be better adapted for planting in shallow-rooted or hardpan sites?
Would they be better for shallow sites? That's an excellent question. I don't know that a shallow-rooted fibrous root system would be better for a hardpan site. I actually think perhaps the plant that has the taproot and maybe the sinker roots could be a better, could be if they can reach the deeper soil horizon. But I think maybe it depends on the perspective and the context. I think if that hard pan is not penetrable by roots, then could the fibrous one do well? I think it could. And I think really the reason I'm saying that is less of the morphology, but more of the fact that it seems that these plants are able to tolerate drought, not just resist drought or avoid drought, but they can tolerate it. So that's a good question. I think I need to do a little more work to try to get a better answer, but I think it really depends on the context of if the roots can penetrate past the hard pan, because these plants are known for having a really strong aggressive root system that other tree species may not be in that same ranking, if you will.
One of the things that we talked about is the difference in morphology of the asexually propagated plants may make them more amenable to transplanting. Right. So we have yet to look at that.
Right. Yeah, they could be that any of the issues that might have led to generalizations with taproots could be alleviated with those fibrous root systems.
More questions?
I have one more quick question about the propagation. What happens if I now just make a cutting and put oxen on it and put it into media in my greenhouse? I haven't done it. I was going to do it this spring just to see if any of them would root. Just, you know, last year's growth, cut off and put right now is a good time, I think, for apples at least, or other things to do that.
So the question is looking at just other forms of vegetative propagation.
Right. People probably tried it. I just wanted to know from you if you've tried it.
Yeah, so it's definitely been tried. I've tried it myself. It's usually unsuccessful. Some of the pecan literature suggests that dormant hardwood cuttings could be possible, but the rooting percentage is really low. And I have not seen any evidence, and nor have I been able to do it myself with softwood or semi-hardwood cuttings. Cuttings really seem to be more difficult. And the benefit to like that modified layering technique is not only are we taking advantage of exogenous auxin, the plants are still connected to the mother plant, and but they're also juvenile tissue. So because they're coppiced, it's juvenile tissue. So those are all kind of added bonuses to, in the general sense, thinking about which factors might help boost rootability. So you kind of check all the boxes with that technique.
Thanks.
Yeah, absolutely.
Hi, Brandon. Wonderful talk. Nice to see you again as well.
Nice to see you, Paul.
I just I wanted to ask about like regarding the Shagbark hickory with its very poor growth rate as a seedlings. I was wondering if this might have something to do with its ecological niche, like maybe they're very like shade dependent when they're young. Did you look into that possibility? And another question was also kind of related, but possibly the role of microvizor in microvizor associations in transplanting or even like the growth of seedlings.
Yeah, that's a great question. So most of the hickories, if you look at forestry literature, are known to be very shade tolerant. So these are plants that will have to compete in heavy shade. And you're probably right there that part of this sequence is being able to survive that period. One way that you could look at it is they put all this energy into the taproot. Hickories are notorious for being able to resprout. And so deer browse is a big problem in a lot of the habitats that you find them. So seedlings could be browsed back many, many times. But if they have their energy stored in the taproot, they're more capable of being able to resprout and try again. Or like you've mentioned with shading, waiting for their moment for light to open up in the canopy and they can go forth. You can try them in shade or in the sun in a production setting, shagbarks in particular. It doesn't change the growth, not that much. You get a little bit of shoot extension, but not, they still go through that period, episodic growth. And, the differences are centimeters, if anything at all. The second question about associations, yes, I have not done any work with associations, mycorrhizal associations, but I know for a fact that Carya liceniosa, the kingnut hickory, it is known to have an ectomycorrhizal association, and I can't think of which organism in particular, but I think with most plants, whether we have it documented or not, there's probably a bunch of associations that are going on. However, I do know there is one noted example for the Carya liceniosa, that riparian species.
Thank you.
Yeah, great questions.
So are we up, Don? I think so. Okay. So thanks so much, Brandon. It's always a pleasure.
Thank
And I really appreciate giving your full viewpoint here today.
Wonderful. All right. Thank you all very much.
Take care.
Thank you. Bye.
Bye-bye. Take care.
This has been a production of Cornell University, on the web at cornell.edu.